right. Well, it wasn't too long of a break, so hopefully you remember a little bit of our cell biology. Um, or don't, that's fine. So this was my conclusion slide from earlier, looking at cyclin C, how it is apparently localized to the mitochondria in MED13L mutations. This has both transcriptional and mitochondrial impacts, specifically mitochondrial dysfunction. So a little bit of the summary from before. 11 of the 12 cell lines that were examined had mitochondrial fragmentation, 9 of which had 3 times more fragmentation than the control. More importantly, the genotypes associated with the most mitochondrial fragmentation were those that were deletions and were N-terminally located along the coding region. The same was also true for mitochondrial dysfunction in terms of ATP production. The exact same genotypes were implicated here, deletions and N-terminal location. And something that I briefly touched on at the very end, and these studies are currently ongoing, is that 5 of 5 cell lines tested aged prematurely. So again, one of the hot topics in aging biology is premature senescence. These studies were done once and are currently being done again. A random little side note, these cells are actually in the incubator right now at about halfway through their lifespan. So this is right when we need to start targeting for early premature senescence. So on Monday morning, Randy, I will be going to go treat cells with anti-senolytics. So future directions for the work and the drug repurposing studies that you all have been waiting for would include testing small molecules and compounds that have already been FDA approved for patient use to help with some of the molecular phenotypes that we see. For example, as part of its emergence with aging studies, we have senolytics, is what they're called. They're anti-senescence drugs. They have been produced to specifically target senescent cells and induce cell death in those specifically. Like I mentioned, this treatment would be administered toward the end of the cell's life as to help potentially with the longevity of the cells. In addition, we can treat cells with ROS scavengers to help mitigate the damage from the mitochondrial dysfunction, especially since this fragmentation has been typically seen to lead to extensive ROS, and then we also quantified this using flow cytometry to show that, yes, ROS is elevated both cytosolically and at the mitochondria. And lastly, we could combat mitochondrial dysfunction with different drugs, such as a chaperone inhibitor for cyclin C called PFT, which blocks cyclin C mitochondrial localization. And all of these treatments could actually be supplemented with mitochondrial biogenesis drugs, such as those that stimulate the AMPK signaling pathway, and all of which are under work right now. So one of the first drugs that I tried was mitoquinone, MitoQ, CoQ10. It's an over-the-counter supplement to help with excessive ROS in cells. It's an antioxidant and is a modified version of CoQ10. It actually has modifications in its structure that allow for cell permeability. And the way it works is that it donates electrons to various free radicals. You can see oxygen here, the hydroxy radical. It helps mitigate DNA damage that's typically the cause of reactive oxygen species. ROS can also damage other cellular components. So this would help reduce the oxidative stress or the oxidative burden on these cells. And it can actually cycle and scavenge more than just one free radical. So currently, a few baselines have been tested in terms of concentration of this drug to add in a cell culture model. I actually found out very quickly that too much of this drug has an adverse reaction. So it causes more ROS if you have too high a concentration of CoQ10. So once I found the appropriate concentration to work with, I was able to treat cells for a variety of times, so whether that's 72 hours, so a short time course, or over the lifetime of the cell. I have a couple different patient cell lines here. All of the cell lines had elevated cytosolic ROS. So first I was looking at DAG, cytosolic ROS. And you can see that with this treatment of MitoQ, that there is a decrease in all three of the cell lines that were tested in terms of overall cellular cytosolic ROS. So on that last slide, I was showing cytosolic ROS. Obviously, this has to be repeated with mitochondrial-specific ROS, since this is a mitochondrial-specific drug. The patterns were exactly the same that I saw for the cytosolic ROS. This was using MitoSox as my marker here, and was performed as the same flow cytometry assay as before. So this confirmed that MitoQ was also successful in mitigating the mitochondrial ROS, as these patterns were similar to those of cytosolic ROS. I was able to treat these cells for the entirety of their lifespan without any adverse reaction. And you can also see that in control cell lines, there is no adverse effect. Next, I wanted to target mitochondrial biogenesis. So one of the first drugs that I tried was metformin, which has a couple different proposed mechanisms of action depending on the literature that you look at. The one that I'm going to mainly be talking about today is through the direct activation of AMPK signaling, which in turn activates PGC1 alpha, which is the master regulator of mitochondrial biogenesis, leading to an upregulation of NRF1/2, the mitochondrial transcription factor, TFAM, that helps with overall organelle biogenesis. So a direct way to add a drug to these cells to help produce more mitochondria. So in terms of the results for this, I was doing the seahorse metabolic analysis in both untreated and treated cells. For a couple of these one cell line here, we had a very drastic increase in the mitochondrial output, showing that this drug is doing what it's supposed to. The other ones were a little bit less drastic in terms of their increase. So potentially this drug is better in combination with something like a chaperone inhibitor like PFT to help with cyclin C interacting with the fission factor, so stopping mitochondrial fragmentation at the start, and then supplementing with something like metformin, we're able to stimulate biogenesis. The last drug that I tested was elamoprotide. Its mechanism of action focuses on the mitochondrial inner membrane, so a little bit of cell biology here, specifically binding and stabilizing cardiolipin. So working our way through this diagram, you can see these invaginations of the mitochondrial membrane are known as cristae. Randy pointed these out earlier. They are where the electron transport chain supercomplexes lie. You can see that in a pathological model or an aging model, there's less surface area for these electron transport chains to be set up, supercomplexes to be set up, so there's less ATP production in these cells. However, in the case of treatment with elamoprotide, the small molecule, this binds to cardiolipin, which helps stabilize the structure of the cristae even when they were pathologically affected earlier, and restore ATP production. So again, I wanted to do mitochondrial output assays, so looking at seahorse. These are still ongoing, but I do have some preliminary results. You can see that this is the same kind of graph as before. There is an increase in mitochondrial function for all of the cell lines that were treated. However, there was a bit of a negative reaction for the control cell line. Don't necessarily have an answer for as to why right now, but we're more so looking at these bars that we have an increase in mitochondrial function when we're applying elamoprotide. So the whole goal with these studies is to apply different drugs to these cells and use the mitochondrial dysfunction as a biomarker that we can look at to see if we are able to restore mitochondrial function. Also testing different compounds of drugs. So most drug repurposing studies are different compounds and cocktails of drugs that are mixed together. More so a multi-pronged approach to this. But again, a lot of these are preliminary, and that's what my current postdoc is on, is looking at different drugs that I can add to these cells and look at their mitochondrial function. And that is it for right now.